I want to give a bit of seasoning for Andrea, who comes to us from California, in particular in Berkeley. So I met Andrea because I crashed a amazing experimental rotation they were doing in the MBL in a physiology course this last summer. And I was just really struck and blown away by how Andrea was doing these like incredibly technical, like nowhere else in the world could these experiments be done. But like this was just like a very casual thing that you would present, like crazy movies, as you will probably very casually do over here. And I really appreciate all the pedagogy and generosity that you brought to every like human interaction at the whiteboard or with students or with fellow scientists. So I think that energy will be mustered here today. Thank you for coming.
Good morning, everyone. Like Gabe said, I'm Andrea. I'm in Fernando Garcia's lab at UC Berkeley. I'm a grad student. And first, I want to say thank you to all of you and organizers. This conference has been so fun and energizing, and I'm just having a great time. Today, I'm going to talk about a project that is very much still happening. We had a crazy idea and it worked. And now we're going to have data that I couldn't imagine. So I wanted to bring it here today because this question of what is the right model is something that is coming up sooner than I thought. And I thought this was a room full of people who would see ideas that I wouldn't even think of. So toward that end, if at some point in the talk you're curious about something or something doesn't make sense, I built in a lot of time for questions. And I tested this on my group, but they're a really bad representation of this audience. So please just jump in whenever you're curious. All right. So I study transcriptional regulation.
Sorry, in the center.
In the center?
Yeah, this is perfect.
Oh.
You can slide the board over when you're done. So.
Okay. Excellent. So like this is my boundary. Okay. I study transcriptional regulation. So let's just start by talking about what happens when a gene is transcribed. So this is our gene. And when it's transcribed, RNA polymerase comes to our gene and elongates across it to create a nascent mRNA. So this process is controlled by transcription factors. So this is our transcription factor, which bind to another region of DNA that doesn't code for a protein called an enhancer. And the combination of interactions between our transcription factors and this non-coding DNA, oh, I have multiple colors, yes. Influences this transcription rate. I don't know how it influences the transcription rate, and that bothers me. And when I say how, I don't just mean like what are the molecular players, but what are the key limiting factors of transcription and how do transcription factors help you overcome this? And I think one reason why this picture doesn't seem to tell the full story is because it isn't the full story. Transcription doesn't happen in a vacuum. The nucleus is this deliciously complicated environment where you're surrounded by all sorts of RNAs and other genes and proteins, and we're just kind of beginning to understand what's going on. And so I want to focus today on the fact that when one gene is transcribing, it's very close in space to other genes. This is a second gene and enhancer. And when I'm talking about proximity today, neighboring genes, I mean neighboring in 3D space, because you can imagine also being neighbors on your DNA fiber. So it's been shown that genes that are close together can influence one another's transcription positively. So if I have a lot of transcription happening over here, you may also see transcription here. So the general conceptual model for how this happens is that whatever the enhancer is doing has some sort of spatial effect. So one picture is that you're creating a hub of resources that are important for transcription. And these resources can be shared by everyone in the neighborhood. So this picture matches the existing experimental data, but it doesn't really let me in under the hood to build intuition of what's going on. But it's very tantalizing, right? Because it says, oh, there is something that I'm missing. There's something limited, like maybe I have many reactions happening in a row that all require some ingredient that there's competition for, and I want to be greedy and gather it around. There's other hypotheses too, and we don't even know that it comes from the enhancer. It could just be that transcription begets transcription, and once I start, everyone around me also starts. So, yes.
Characteristic decay of these correlations in space.
That is my mission. That is what I'm trying to learn. Yes, you set me up perfectly. So the question is there some characteristic decay of the correlation in space? So yeah, if I want to get a better intuition for how this happens, I think the starting point is I have a wish list of measurements that I would like to make. And I'm going to now throw down a very greedy experimental gauntlet that I, as my own experimentalist, have to fulfill. Okay, so here's my wish list. So I want quantitative readouts that I could use to benchmark my eventual amazing model that I will build. We will all build. And then specifically, I want to understand the correlation between the transcription rate at both genes. And if I'm being really greedy, I want the length scale. And I also want the time scale of that correlation.
That's all.
Thing is, it works. The other thing that would be nice is if I had knobs that I could turn. So once I characterize this behavior, if I could change something significant about the system, and then I can test whatever intuition I've built. So there's probably a million knobs you can think of, but two that come up is the concentration of my transcription factor, and if I can vary the number of binding sites for my transcription factor on my enhancer. So essentially, how strong is my signal and how readily can I read that signal out? Yes.
Do we know anything about how genes cluster in space? Are related genes clustering together in space, or is it random?
In terms of related,
In terms of function, or it could be anything, but.
So as far as I know, I haven't read a paper where related functional genes cluster in space. That doesn't mean there isn't one. In terms of transcriptional activity, there have been some papers that look at like simultaneous imaging of DNA and RNA in fixed tissues, and you see that there seem to be bubbles of correlated transcription activity, but functionality, I haven't seen anything.
So as far as you know, it's random. Genes are a little bit randomly collected in spatially.
Yes, but I see the picture you're pointing to, right? I say, oh, I'm a fly and I'm making my head, and all of my head genes are in this little bubble of the nucleus. Let me put them all here and turn them on. That is a very exciting idea. And yeah, I don't feel like I know the answer to your question affirmatively enough that I can say yes or no. Yeah. Okay. So yeah, today I'm going to talk about an experiment that we've basically, right now, we're validating it. That gives me access to this. And then if I have time at the end, I would like to pick your brains a little bit about how to formalize my intuition in terms of theory and probe this more rigorously. So the system that I'm doing this measurement in is the fruit fly embryo. So Madhav set me up a little bit yesterday talking about the fruit fly embryo, but here's a recap. So eyes not drawn to scale. So this is our embryo. Ultimately, it will grow up to be a fly. And the cool thing about the fruit fly embryo is it's a system where turning knobs is really, really tractable. So for instance, all of the nuclei in here have to know where they are. And that information is encoded in concentration gradients of morphogens, transcription factors. So just position in the embryo is the x-axis here, and you have built-in biologically relevant concentration gradients. So we get that. And then the genes that read out these transcription factors, their enhancers are relatively well characterized, so we can delete or even add binding sites. So that's it. Knowing about the knobs is the easy part. Doing the experiment now is the fun part. So the next thing we need is two genes that are coupled in space that I can measure easily. And the dream experiment is, let me just take the same gene, make two copies of it, and measure how correlated its transcription dynamics are. And the amazing thing about fruit flies is that they do this for me. So if I take one of these nuclei and zoom in a little bit. So in the early embryo, in the fruit fly, so much is happening all at once that it leads to this very unusual cell cycle. So not only do the cells need to divide very rapidly with 9-minute nuclear cycles, but they have to produce a ton of gene product because they're having this constant conversation to say, this is where I think I am, what do you guys think? So they have to read in transcription factors and then produce more transcription factors. And what ends up happening is your genome is duplicated prior to the onset of transcription. I have a physics background, but I believe it is usually the other way around. So this is one strand of DNA, and here's your sister chromatid that has already been created at the time transcription begins. So, every gene that we look at is actually two identical copies of the gene that are tethered together. So I'm not the first person to think this is an interesting thing. And there are two previous studies that have looked for this. And what's interesting is that they have conflicting results. So I'm just going to touch on that quickly and then jump into how we're going to hopefully resolve that distance with live measurements. So the first of these studies was done in the 70s. So it's electron microscopy data where you identify pairs of chromatids and then count how many nascent mRNAs are on each one. And so if these are the nascent RNAs, I can see based on how far along the gene they are that like these were created at the same time, these were created at the same time. And you get an idea of the history of how correlated expression was. And so from these EM experiments, you can say, if we just arbitrarily decide this is gene 1 and this is gene 2, so the amount of mRNA on gene 1, can you guys see behind the podium? Whoa.
ADA compliance, so.
But it doesn't go down any further than that.
Doesn't go down any further than this. This is the best we can do.
So what they saw from this experiment is like in an obscene degree of correlation. They looked at 68 pairs of genes, and I was going through the table, and almost everything is above like 0.95 correlation coefficient, and the lowest one is 0.72. So this says sister chromatids fire together, right? And then in 2013, there was a study that used single molecule fish. So this is a technique where you create a fluorescent probe that's conjugate to a nascent RNA sequence you're looking for. And essentially, the more nascent RNAs you have, the brighter fluorescent signal. So it's a different way of counting how much RNA you're making. So if this is our gene, in this experiment, you would see two bright spots. And you measure the intensity of them, and you compare them to one another. And in this experiment, they saw uncorrelated transcription. So my awesome rotation student and I went through all these papers when we kicked off this project. And so there's two hypotheses here, right? Either transcription has dramatically changed since the 70s, or these experiments are actually measuring different things. And the trick is that pairs of chromatids are dynamic. So if this is my two chromatids, sometimes they're separated, and sometimes they're virtually co-localized. And this goes back and forth on a time scale of a few seconds. So, but each of these experiments is sensitive to a different one of those modes. in the Miller spreads experiment, the way that you can be sure two sister chromatids are two sister chromatids is if they are very, very close together. In the single molecule fish experiment, microscopy generally is limited by the diffraction limit, the smallest thing you can make out. And so when they're co-localized, you have to throw away those data points and you can only look at the far apart ones. So there's potentially a unifying hypothesis here where both are true within the life cycle of one gene. Do you know how I am on time? I forgot to start my time.
Good. 11 minutes.
Okay. So what we're trying to do now is resolve this difference using dynamic measurements of transcription. So that's using a system called MS2. So this is our gene once again. And we put these, the sequence stolen from a bacteriophage that when it's transcribed forms this stereotypical loop structure. And so then these loops are at the end of our RNAs. And we engineer our fruit flies so that they will express a fluorescent protein that binds structurally in a structure-specific way to these stem loops. So every RNA comes with a fixed signal, and it's essentially a live version of this single molecule fish experiment. So in our nucleus, we can just measure the intensity of an MS2 spot. And you get a time series of signals that correspond roughly to your transcription rate at that time point. There's some convolution with how much time it takes for the polymerase to cross the gene, but we've actually developed a method that lets you go backwards and deconvolve to transcription rate. So we can think of MS2 as more or less the same as transcription rate with some time lag, which is actually useful for our analysis. But the thing is, this transcription spot, as we know in the fly, is actually two sister chromatids very close together. So in my group, people have been working with MS2 in the fly for more than 10 years. And previously, with the spatiotemporal resolution we had access to, it made sense to just sum the two chromatids together and treat it as one effective locus. But I've told the whole story in backwards order. We had this idea first and then developed the experiment. What actually happened was this summer at Woods Hole, one of our students said, what happens if we just zoom in and go really fast? And we did, and we saw amazing things that, so slowly, this is a very wonderful time to be working in fluorescence microscopy. Like photon detectors are so good, fluorophores are so good. And so we had added all this juice technology, but we were still using our experimental biases from before we had it. So like being shaken loose of those freed this awesome experiment. And so the idea is, if we just zoom in and go very, very fast, can we individually track these sister chromatids and get their individual MS2 trajectories? I'll show you those movies in a second, but before I do, I just want to give an intuition for the length scales and time scales so that we understand why this experiment works. Let's see. So in terms of time, so this is a few seconds. The time it takes for our polymerase to cross the gene, so we can think of this as the memory time for our MS2 signal, is a few minutes. And then the time scale of these fluctuations in our transcription rate. So you have these long, so again, a few minutes periods where the gene is generally on or off. And then within that, you have smaller fluctuations that we're still kind of learning how to understand that are on the time scale of around 10 seconds. In terms of spatial resolution, when the chromatids are farthest apart, again, this is something that we're still measuring, but I would say the number is around a few 100 nanometers. At their absolute farthest apart, but generally they're a little bit closer than that. Now, the diffraction limit of our microscope is around 200 nanometers, but the diffraction limit only points to the information that's present in just the picture, but we know more than that. We know it comes from 2-point sources. And so that gives us an experimental spatial resolution. I guess I can just write. My friend and very close collaborator, Jovan, is literally finding this number right now today, but he believes our spatial resolution is around 45 nanometers. And in terms of our temporal resolution, we're imaging at 3 hertz. So like our sampling rate is faster than all of these timescales. So that's what you're about to see. Oh, I have to do all of this. That makes sense. Power on. And then Aaron down. I'll do both of them for back.
Yeah, drama. Drama's important. Dun dun dun. Drum roll.
Okay. So what we're looking at now is a section of cells inside of a developing fruit fly embryo. So each of these blobs here is one nucleus, and the white spots are MS2 spots.
Crazy.
And then just to give like a more zoomed in picture, here are the 2 sister chromatids coming apart and back together.
So in the chromatids themselves are like on the order of a few hundred nanometer, like the bright, the spot that we're seeing itself.
The spot that we're seeing, I think, would be on order 100 or less nanometers, and then the distance between them would be up to a few hundred. Our rotation student thinks he's seen them a micron apart, but I need to go home and look at that movie. And so the idea here is with these movies, we can look at not just one MS2 trace, but we can look at two sister chromatids and see how correlated their expression is with time-dependent information. The one thing is, in order to do that, you need to be able to consistently register the identity of each chromatid because they overlap. So we actually figured out a way to do that based on the timescale of this is much faster than the timescale over which this happens. So you can come up with a probability of who is who. And we didn't think that this would work. We came up with the idea at 5 a.m. and then it worked. But yes, so it's still in progress and we're validating it on simulations. But you can see the pink and the green label correspond to the confidence of which one is which. And in the plot on the right, all the little dots are the samples of the distribution of the, I can get more into that if people are curious. Am I totally out of time?
3 minutes.
3 minutes, okay. So with that small time window left, I want to come back to the big questions that I started with, which are how do genes couple in space, and how does this understand what's actually happening when we come down to transcriptional regulation? And we have all of this information. We can look at the correlation of these genes in time and space. We can also measure the distribution of the distance between the two chromatids as a function of time. And the ways that I've thought of to model all of this seem like I'm leaving a lot of cool information on the table. So my very naive approach, would be to say, okay, I have my gene, and I have a neighboring gene. And let's just say I transcribe if I have an activator bound to my enhancer. This is a version of the occupancy hypothesis that a lot of people use in transcription field. So I can let's just start with equilibrium average distribution and say in the free energy minimizing distribution, how often do I have something bound to my enhancer? And as I tune my transcription factor concentration, I can tune how much mRNA I make on average. So I can imagine adapting this model to say, well, now all 4 of these are my binding sites, or these 2 are mine, and these 2 are mine with some radially decaying coupling constant. And I can predict, okay, do I see a different input-output function now? And based on our measurements of this for like chromatids combined, you could maybe limit some of these parameters. But we have all this delicious dynamic data, and like this feels like just like a first step, and I'm not sure what the next one is. I can imagine writing a kinetic model where I think about transitions between all these states, but I'm not sure if that's the best way to get a foothold into the intuition here. So now or later, people who maybe are more adept at this sort of thing, I would love to pick your brains. And I guess the other thing of note here is this trajectory, like inside of a living organism. We don't think anyone actually has measured this before. We kind of did it on the way to this other thing, and Herman was like, no one knows this either. But I don't know any polymer physics, and I think there's like very cool stuff here. And I would love to talk to someone who does, and I know you exist. So with that, I guess I'll wrap up and see if there are any questions. Yeah.
I need to know, 'cause I feel like I missed the answer. What is the answer? Are they correlated? Are they not
Oh my
correlated?
gosh. So this is your fault for making the conference this week and not 2 weeks from now, because we will know next week.
Okay. Sorry. So these genes are the same, right? So I guess in another universe or in your next career, I'm also kind of dying to know, but what if they're not the same? What if 2 genes are close together, but like back to the question of, are all the head genes in the same space? Is this pattern because they're the same copy? 2 alleles of the same gene?
Yes, so we can do that experiment as well. I think it's a little bit trickier because like we get for free the fact that the alleles are, the 2 sister chromatids are glued together. If you want to look at coupling of other genes, you like the standard way to do it in the field is to like tether them together by altering the DNA, which obviously is like a bit more invasive. But yeah, we're working on actually a live version of that experiment. This is the same platform. It's a really good question. In the back.
Well, just to kind of talk about that too, this really made me think of all the older expression systems where we really crank up the expression of like GFP tag proteins by using super high promoters and whatever else. So how much are we actually changing other genes without even thinking about any of this?
Yeah, it's a great question. Like the nucleus is like so interconnected. And I have this totally wild speculation, intuitive picture of how, like it's this giant spider web where like you touch something over here and then suddenly the nucleolus is mad. And this is, yeah, it's very beautiful and terrifying. In the back, yeah.
Presentation. So I have a question about these videos that you showed and like the probability distributions. So kind of a 2-part question. One of the questions is just if you give us like a little bit about the methods and how you did this. And then the other question is, I see that kind of when the white splotches merge, we have this like mix of the pink and green afterward. And I'm wondering, is it attempting to track the identity of the unique particle or just separate like the difference? Because I see like the pink and green merges and then they go back, right? We have pink and green that is mixed and then it becomes pink and green again. So is it tracking difference or tracking like true identity across?
That's a great question. So we can track the locations with, like I said, a 45 nanometer precision. That's a different thing where we just say, like, we know that this is 2 points and like, so like, where are the most likely centers there? So the points are spread out here just to show how many there are, but like they don't correspond to the spatial location. In terms of how the thing actually works, this is actually my favorite part of the project. This was our crazy idea. And we came up with like plans B, C, D for if this doesn't work. And we tried all of those first and none of them worked. And then we were like, well, I guess we can do our crazy plan A, and it worked like overnight. So, oh, I erased the thing I need. Okay, so let's say you have, these are your two spots. They're separate, and then they come together and they separate again. And let's say I'm tracking the intensity of each of those spots. Let's see. Let's say I've arbitrarily labeled this one as red. And I get my trajectory of red spots here. Then I have this other spot where I can say, all right, this one's intensity is this much. Now it's this much. Here, I can't measure the individual intensities, but I can measure the sum intensity. So I guess not perfectly drawn to scale. And then they come back apart. I'm going to draw an extreme version where this is very obvious, so if they look like this, you know, based on how slow varying the MS2 signal is, since it has the whole memory of the last few minutes as polymerases were crossing the gene, that it's impossible for this lower spot to not be the red one, or this, like, because this upper one couldn't have lost this many this quickly. The key parameters here is like how quickly do they overlap versus how quickly we measure and how quickly the signal varies. And like, because they come apart and go back together so quickly, even if they're co-localized a lot, we still get individual samples. So far we found that it works, but we're running simulations to see if there's failure modes we haven't thought of. Does that answer? And I think there was a question over here too.
The transcription factors, do they affect the mechanics of the DNA at all? Like, do they increase bending rigidity or something?
So I know that the transcription factors are sensitive to the curvature of the DNA, the specific ones that we're using. I strongly suspect that they influence the mechanics. That's like a whole subfield within my field, but I don't know the exact specifics of how they do. And then there's like some hypotheses that they participate in like displacing nucleosomes from the DNA, which relates to the mechanics, but is also about the packaging. Yeah. Do you have a like intuition for what that would mean?
We can chat after.
Awesome. Yeah.